After following the passage and putting cells back into the flask, place them in the incubator before starting cell seeding. First, we want to prepare our well plate. Ensure that you UV any scaffolds, syringes, and needle tips before using. Then place scaffolds inside of your 12 well plate. Next, we need to mix ourselves with our hydrogel, whether you're using Matrigel or Geltrex. First, you want to grab a 2 milliliter tube to place your cell-laden medium and hydrogel in. Compositions will be dependent on trials that are being taken after ensuring you got the right concentration. Take the mixture to the vibrator and vibrate 2 to 4 times. After cleaning it off again, we need to place it into the syringe to seed onto our scaffolds. First, place your needle tip onto your syringe and then use the 200 microliter pipette to place your cell hydrogel mixture into the syringe, and be sure not to place your mixture directly into the needle tip to avoid clogging. If your needle tip does clog and provide it, you have an extra needle tip. You can remove the clog, needle tip, and use a 200 microliter pipette to transfer the cell hydrogel mixture from the clogged tip back into the syringe, and then placing a brand new tip onto that same syringe. Then reinsert the white piston into the syringe if you're ready to see it again. When seeding the cells, try to be as gentle as possible as to not lose any of your mixture, or place copious amounts of your mixture onto the scaffold. Holding the well plate in one hand and the syringe in the other allows you to trace the scaffold channels with precision, avoiding loss of your cell mixture. Following seeding, place the well plate into the incubator for 10 to 15 minutes if using Matrigel, or no less than 4 hours for Geltrex. Rehydrating every 30 to 45 minutes. When your hydrogel is done polymerizing, overflow your cells with 2 milliliters of medium and place back into the incubator.